presentation?
Yes.
Okay. Okay, so good morning, everybody. So my lab works on personalized medicine approaches using large-scale data from human cohorts that we collect. We've been working in this area for quite some time. The first project we published, actually it's the Zevi Cell paper in 2015, on studying the question of nutrition. We collected a cohort of 1,000 people. We called it the Personalized Nutrition Project. It mainly focused on using continuous glucose monitoring for looking at blood glucose levels in people after they eat meals, because that's important for weight management, for diabetes, and it's easy to measure. So this study was published a few years ago. I'll just summarize the key results. We showed on a scale of 1,000 people that different people have very different blood glucose response to food, as you can see here in the response of 4 people to bread, while the same person has very similar responses on different days eating the same meal. We showed that using gut microbiome and other clinical parameters, we could quite accurately predict the response of meals of participants. And in a small cohort of 26 people, we showed that we can take people who are even pre-diabetic, that have these spikes in glucose levels, and if we give them a diet with exactly the same amount of calories, but tailored by the algorithm, we can fully balance their blood sugar levels. So when we completed that, the question was, what happens if people follow this for the long term, which is something we tested in a randomized clinical trial just published in Diabetes Care a few months ago, where we took 200 people with prediabetes. We randomized them into either our algorithm diet or the standard of care diet. We mainly looked at changes in blood glucose parameters, because that's the main issue with people with prediabetes. The study was half a year of intervention, 1 month prior of profiling, and then another 6 months of follow-up. We used CGMs, glucose monitors, for the entire duration of the study. And we also looked at, as secondary outcomes, at different lipid profiles and blood pressure and other metabolic parameters, some exploratory markers of microbiome and metabolomics, and people logged all their food intake during the study. So here's one participant from the algorithm diet. You can see prior to the intervention a lot of spikes in glucose levels in this person, but in the 1 month after the intervention, we virtually eliminate these spikes on the algorithm diet. And for the entire half a year of intervention, you can see that there's a very notable reduction in glucose levels. And this is another participant where, again, you can see spikes in the pre-intervention period, and then a much significant reduction in the intervention, and another participant, and another one. And even at the individual level, when we look at all participants, then their response to meals in terms of glucose levels was high prior to the intervention and much lower during the intervention on the meals of the algorithm diet. And this is true at the individual level for the majority of the participants. In contrast, when we look at people on the standard of care diet, this is the Mediterranean diet used to treat people with prediabetes or diabetes, we see that the pre-intervention period and the intervention period, at least by eye, seems quite similar. And this is true for many participants. And when we summarize, and this is the key primary outcomes of the study, we see that on the standard of care diet, after half a year of intervention, there is a significant reduction in hemoglobin A1C, but in the next 6 months of follow-up, there is a reversal back to baseline. In contrast, on the algorithm diet, we see double the effect of after the intervention, and very nicely, we see that this effect is also maintained in the next 6 months of follow-up. And we see here on the right, also a 70% reduction in terms of the CGM, the ability to reduce the spikes in glucose levels. So just to put this in more context, the NIH did a study on 3,000 people with prediabetes, considered the gold standard in the field, the diabetes prevention program. You can see here the line of placebo, the line of metformin that UN also just discussed, and you can see that it's actually doing in between their third intervention, which was a lifestyle that also included physical activity and caloric restriction, both of which we did not do here. We just did a dietary intervention. And what was nice is that we got an effect that is even greater than what the DPP got just by changing the diet without adding physical activity or doing caloric restriction. So why am I showing you also this? Because the NIH also followed these participants for another 4 years, and they showed that their lifestyle arm actually reduced the conversion from prediabetes to diabetes by about 60%. So we're hopeful that because we got an effect that is even bigger, then this actually would lower the conversion to diabetes by an even additional factor. So, this for us concluded a 10-year journey from an idea to development of the algorithm in that 2015 paper and up to now what we think is a demonstration that this algorithm, if you follow it for a long term, can be a treatment for people with prediabetes. And we have another paper in review showing that this is also effective for people with type 2 diabetes. So when we finished this study, we wanted to ask a broader question, which is what would happen, or can we use these population level human cohorts to tackle not just human nutrition, but even more broadly human disease and develop new therapies. So we initiated a few years ago what we call the Human Phenotype Project. And really the goal is to address this very concerning problem, which is if you look at the cost or how much spending we have to do on the development of each new drug, you can see that this has actually been going up exponentially. This is a log graph of costs, and you can see that bringing new drugs to the market now costs more than $1 billion, bringing just a single drug. And if you look at this graph recently, you can also see that it may be plateauing. And some people consider that this may be because of the use of genetic data in the development of drugs. And it's been suggested that every time that you integrate genetic data, then you may reduce the cost by a factor of 2. So our hypothesis is that because genetics is just one component and it's also a predetermined component that doesn't change, in contrast to other omic data sets, then our hypothesis is that by integrating deep multi-omic data, we may be able to drive the cost of these new drug development much faster. So as a first milestone, we set out to collect data from 10,000 people. We are also following them longitudinally, and what happens is that some people, when they're recruited as healthy, remain in their healthy state, some progress towards disease, and some then start to develop disease. And the hypothesis is that much before clinical symptoms appear, you will see some mark in the molecular data, and we may be able, from the baseline samples, to detect which people are progressing towards disease and which remain healthy, and that will lead to diagnostic biomarkers and some that may also be therapeutic. In addition to this healthy cohort, we're also collecting, in collaboration with clinicians, data on various disease cohorts from oncology, cardiovascular disease, and so on. And this project has been going on for the past 3 years. We have over 15,000 people who registered. We already profiled more than 8,000 people, and we're continuing this profiling purpose. Some hundreds of people have also returned for a second visit. So we have 2 samples of them after 2 years, and hopefully we'll be able to expand this much further to a larger cohorts and to cover also more disease areas. What enables us to do this is technology that we've developed over the years to really streamline the process of data collection. We call this low-cost phenotyping. And just to put the project in perspective, of course, we're not the first to profile large human cohorts. The way I like to think about this is on 2 axes. On one, you have the number of people being profiled. On the other, you have kind of the quantitatively how much you're profiling them. So we have, for instance, there's electronic health records that we also use in Israel, databases that sometimes even go to millions of people, very impressive. But on the y-axis, in terms of how deep you're phenotyping, they don't have molecular data, so they're typically not profiled very deeply. Companies like 23andMe and others have data on millions of people in terms of genetics, but they also don't have much more molecular data. Closest to what we do are biobanks. The most notable example is the UK Biobank, 500,000 people, tracked now for 15 years. They have genetics, they have a little bit more molecular data, but also not that much. And so we thought there's a real interesting spot in this space to occupy, which is our 10,000 person cohort, what we call the 10K project, where we can really profile people very deeply. And by very deeply, I mean that every person who comes to the Weizmann Institute undergoes physical tests that include assessment of the cardiovascular system using ECG and ABI. We connect them also to continuous glucose monitors to look at their metabolism. We get their medical records, so previous diagnoses. We use ultrasound to look at fat in their liver. We use DEXA to look at full body composition and bone density. We use sleep monitors that people go home with to assess the sleep quality, and we get a lot of very interesting analog data on sleep quality during the night. People log their diet, so we know their dietary intake. We biobank both serum and live cells, PBMCs. We record their voice. We use wearable devices to track their physical activity. And then on the molecular side, we do essentially all of the profiling that is known, that we can do at low cost and high throughput. And this includes full genome sequencing, RNA sequencing, proteomics, microbiome from the gut and oral microbiome, and metabolites from the blood using metabolomics, and a novel immune assay that can assess the entire infectious history of a person. We can synthesize hundreds of thousands of antigens and look at the antibodies of a person, and with the readout of sequencing, identify everything that that person's antibody recognizes. So we cover many systems of the body. We perform, as I mentioned, many different molecular tests. And of course, then the question is, can we use this data for developing and identifying novel therapies? So I want to mention a few of the approaches that we're taking. The first is we realize that even when you have a healthy cohort, that healthy cohort varies along many different dimensions. So you can use actually variation in healthy cohorts to gain a lot of insights. And just as one example, we used our metabolomics data, which looks at molecules in the blood to predict the chronological age of a person. You can see that these predictions work quite well, but they're not perfect. And it turns out that if we look at people who have a predicted metabolomic age older than their actual age, and we compare them to people who have a younger predicted age, then these groups differ such that people with a higher metabolomic age actually have poor health condition as judged by the fact that they have higher BMI, higher lipids, triglycerides, higher hemoglobin A1C, so they're closer to developing diabetes, higher blood pressure, higher cholesterol, and higher liver enzymes. So we think that now we can introspect these models and find which biomarkers, which metabolites are actually driving the predictions here, and by that identify markers that may be related to accelerated aging, which may also be interesting to examine therapeutically. As another example, we can actually use the fact that we collect so many different types of data on the same people to really fill out, impute, and emulate randomized clinical trials in those cohorts that that may have profiled much less than we did. So the idea, for example, is to use models that we've developed and published, where we showed that using only microbiome data, we can predict with good accuracy levels of some metabolites, even identify which bacteria are driving these predictions. So what you can imagine we can do now is we can take our cohort where we've measured everything, and now let's say that we go to a more shallow phenotyped cohort that, let's say, only looked at microbiome genetics and has medical diagnoses. And what we can do is we can use these models I showed you before to impute and get metabolomic data on this cohort where metabolomic data was not measured. We just use the model that predicts those metabolites that it can from the microbiome, and we get metabolite levels that are not real, they're predicted, but we know the accuracy with which they are predicted. They should be, as I showed you before. And then you can study the relationship between these predicted metabolites and some outcomes. And if the predictions are of reasonable quality, then at large numbers, this may give you a hint to metabolites that may be of interest, and you can do this approach for many other cases. Another approach that we're using is the classical case control study. So when we have a disease cohort, we can compare it to healthy controls, but we're using a slightly different approach. It's a paper that is now in press in Nature Medicine, will come out soon, where we can really just examine a single patient, find matched controls for that patient in our cohort, matched on, say, age and BMI. And then we can look at the metabolomics and, for instance, identify metabolites that relative to this patient's matched control seem to be an outlier, seem to be disturbed. We can identify many of these metabolites, and then using the models from before, we can understand where these metabolites originate from. Are they metabolites that are driven by diet, microbiome, genetics, lifestyle, or other factors? And what we find is that when you take two different people who have the same clinical endpoint, they both had a heart attack, the underlying reason for why they had a heart attack may be different because it seems that their metabolomic disturbances are driven, in the one hand, by more the microbiome, and in the other case, by more by genetics or more by the diet. So this suggests a way to stratify patients. It suggests that the underlying reason for the disease may be different in different people, and that's another way in which these models are quite useful. Another approach is to use all the knowledge that's been accumulating on genetics, so we can import genetic data to compute people who are at high risk versus low risk for developing some disease that do not even exist in our cohort. So say people who are likely in the future or more likely in the future to develop Parkinson's or Alzheimer's, compare them to patients who are at very low genetic risk, and we can identify biomarkers today that distinguish between these people, and these biomarkers may be predictive. They may also be therapeutic. And the final approach I want to mention is the use of microbiome data. So here we can go much deeper, not just to the level of bacterial composition, but really to the level of single nucleotide polymorphism, so changes in just one location in the genome, which we can finally study when we have cohorts of this size of thousands of people. So the idea is simple. Instead of just looking at the composition of bacteria, we map the variation at each position in genomes, and then we compare this variation to variation in a trait of interest like BMI that's shown here. So the pipeline for this is that we apply this discovery pipeline to identify SNPs of interest. We then look at strains, so bacterial strains in which these SNPs reside, and these strains associate with the traits. And what we are working on now is to isolate those strains from specific individuals, and then try and assemble a consortia and use that consortia in clinical trials to see if it has the ability to help in treatment for the trait we're trying to treat. So let me just give you an example. So for BMI, when we do this association, we can analyze tens of millions of different SNPs, and we actually find tens of thousands of SNPs that are significant across many species. You can see the significance level here is very high, but what's more interesting is this volcano plot showing the BMI points that each bacterial SNP explains. It's the signal here is very high, actually higher than what you find for human genetics. So I think these are very interesting signals, and the strength of these signals was what prompted us to really further look into this. And here's an example of what happens sometimes. Sometimes we find SNPs that are actually correlated to each other, and it seems that they actually separate different bacterial strains, where the individuals in one harboring one of the strains is very different from the other. In this case, actually, the SNPs, the individuals that carry the major strain, they actually have a lower BMI. In fact, a very strong effect, 2.3 points of BMI. This can translate into even 10 kilograms of body weight, so a very, very strong signal. Here's another bacteria that explains about 1 point of BMI and another one. And so the idea would be to take these bacteria that seem to maybe have, that at least are correlated to a lower BMI, and to test whether they're causal would be to extract them and see if we can make a formulation of them, give them to overweight patients, and see if we can drive a beneficial effect. We also have such associations in not just BMI, but every trait that we look. So for example, in hemoglobin A1C, we also have very strong associations. So potentially transfer of these right bacteria to patients who have higher hemoglobin A1C or higher BMI may actually help these conditions. And in the final, maybe I'll take 3 minutes that I have, I want to mention another place where we've applied microbiome approaches, this time to try and see if we can treat a disease called atopic dermatitis. So this is a disease of the skin, what's called eczema. It's very prevalent. It affects about 20% of children, about 10% of the adults. And we wanted to see whether changing the microbiome through fecal microbiome transplants can actually have an effect. So we recruited just 9 patients, a proof of concept study. Each patient was his own control. We first gave them placebo capsules, and we then gave them real capsules, where the separation between the dosings was 2 weeks, and we had also another follow-up time point several weeks afterwards. So first, just the clinical effects. You can see here a patient after the placebo. You can see very clearly manifestation of disease on the skin. And after 2 doses of the FMT, you can see very significant clearance of clinical symptoms. Here's another patient, and hopefully you can see the clinical symptoms of disease, and then the clearance even many weeks after we've completed the last FMT. So when the clinicians score this, they see about a 70% on average reduction in the SCORAD here, the score for atopic dermatitis. And we, of course, wanted to look under the hood to see what happens in the microbiome. And we see that when we look at baseline samples before there's any connection between the donor and the patient, we see that for many species, the box plot here represents the distance between the genomes of this species in different individuals. The dots here represent the same calculation, but between samples of the donor and the patient. Of course, there's no connection between the donor and the patient at this stage, so the dots are right where these box plots are. So it shows that the distance between donor and the patient before they're related is similar to just unrelated individuals. After we do the placebo, there's also no connection between the donor and the patient, so we see the same picture. But after we actually perform the FMT, we see that a lot of dots have actually shifted all the way to the left, meaning that the distance between their genome and that of the donor is pretty much identical and very similar. And so we conclude that this is really a transfer of bacteria from the donor to the patient. And when we quantify this, we see that some species actually got transferred multiple times. And so now the idea would be to try and isolate these bacteria and see if a formulation based on these bacteria can actually be treatment for atopic dermatitis. And my final slide is just showing that when you look at the microbiome as a whole in a t-SNE reduction, you can see that samples of the donor cluster together. Samples of patients are somewhere in the space. After the placebo, there's basically no movement of the patients in the placebo. But once we perform the FMT, there's a major shift in the microbiome of the patient. It becomes not identical, but much more similar to the donor. And this actually happens for the majority of the patients that we use. So we think that the microbiome changes here may actually underlie the benefits. And as I mentioned, we're trying to now isolate these bacteria and see if giving them alone may actually be a treatment. So with that, I'll put up the acknowledgement slide of our collaborators and the many people in my group that contributed to this work. And thank you.
Thank you very much for this impressive presentation, and many interesting aspects were mentioned, and accelerated aging and personal